Excellent. Okay, perfect. Okay, and so if anybody, it's never very easy to do this on Zoom, but if people do have questions as I'm going through, please do try and stick up your hand and I'll try and stop so that if anything is unclear, I can explain it. It's not the same as doing it in person, but hopefully we can at least get some way there. So I thought to begin with, I would just, to put us on the same page, give a very brief overview of single-cell genomics and why indeed we should care about it. So when I was a postdoc in Chicago, working with Matthew Stevens at the time, who's my postdoc advisor, I was lucky enough to start analyzing bulk RNA sequencing data. And RNA sequencing data is very powerful technology, but it had a fundamental limitation, and that is that it's an average measure of expression across a fairly large population of cells. So typically you'd have millions of cells as input, and the measure you would get of expression for each gene is an average across that population. Now, in a lot of different settings, that's absolutely fine. If you have a fairly homogeneous population of cells, or in other contexts, if you look at a cell line, for example, it might be okay. But there are certain settings where that average measure isn't going to be sufficient, where you really do need that single cell resolution. You can think of complex cell types like the brain, where there's really a lot of heterogeneity, or in early development, where, for example, the embryo contains a fairly small number of cells and they have very diverse purposes. And there you really want to move from this ensemble average measure down to single cell resolution. And fortunately, over the last decade or so, technologies for doing this, experimental technologies, have come on remarkably fast. When I started my group, it was kind of exciting to get data from 5 or 10 cells, whereas now routinely in an individual experiment, you can generate hundreds of thousands of cells. So you're able to obtain a very large matrix for downstream analyses, where you've got genes on the rows and cells in the columns. And this is basically giving you the measure of expression for each gene in every cell. And it's not only gene expression where this is now possible. We can also measure DNA methylation at single cell resolution in principle at all of the sites across the genomes, and also increasingly other marks, open chromatin, so using ATAC-seq technologies, or even histone modifications using some of the really cutting edge cut and run technologies from Steve Henkoff's lab and others. And as a computational biologist, these types of data are very exciting because they open up a large number of different questions and opportunities that really weren't possible with bulk data. So the most obvious one is that you can take this population of cells and you can cluster it. And there are many different clustering-based approaches, starting with hierarchical clustering, but now increasingly graph-based approaches for clustering cells into different groups, with the aim being in particular to identify rare populations of cells. And I think one of the interesting challenges, as an aside, is how we scale these graph-based approaches up to the millions and millions of cells that data sets are going to become, because actually a lot of the computational tasks that we're able to do with on the order of 100,000 observations become really quite limiting when you're dealing with millions of observations. I think that thinking of clever sampling techniques so that we can actually scale some of these approaches up are going to be important. But not only can we cluster the data, we can also order these cells across so-called developmental pseudotime. So we can order cells transcriptionally from earliest to latest. And there have been a number of different approaches, again, a lot of them based around graph theory, for ordering these cells in pseudotime. And the goal here is that after ordering these cells along the pseudotime, we can then take that and examine how gene expression changes to work out the order in which genes come on and off as you move, for example, through a developmental process. So what do these data look like? Well, a very common way of visualizing these data is in a lower dimensional representation. Initially, t-SNE was used quite a lot, but now UMAPs or other representations are much more common. But the key thing here is that we've taken here actually quite a small data set containing only around 1,000 cells, and we've projected that down into 2 dimensions. So each point in this plot here on the right-hand side corresponds to a cell, and the different color corresponds to the allocated cell type. And then what you could typically do is you would, for example, say, well, this is an ordering from early development through to a middle stage. And up here, we've got some more mature blood cells, and this is a bit of a blood lineage. So we can then compute our pseudotime along here, and then we can ask, what are the genes that turn on during early blood development? But as Jessica alluded to, there's still quite a lot of computational challenges. And I think some of these we put out in this review just a few weeks ago now in Nature Biotech around different concepts for data integration. And so I'd like just to walk you through the first of these and then give a couple of examples and then move on to other types of integration throughout this talk. And the first type of integration that we defined was horizontal integration. And here what you have are 3 independent data sets, for example, in this top row of this little cartoon here. And these might have been generated in independent labs by different groups. And the goal is that we want to combine these data sets together. And the key concept that we're going to use to do this is to use a particular feature as an anchor. And in the context of gene expression data, it's very obvious that that feature is going to be the genes, because we've got the same genes measured across the different data sets. It becomes more complicated, as we'll see, when we move to other types of integration later on. And the first approach that we developed for this, that I'm going to just briefly walk you through because it was published some years ago, focused on effectively trying to remove batch effects to integrate these different types of data sets. So if you've got, say, 2 different labs that have studied cells from the liver, we want to combine them together, these different data sets. And why do we want to do that? Well, it'll give you more power to identify rare populations of cells when you cluster, also more power to detect differentially expressed genes, genes that describe and delineate these different populations. And so when we were first thinking about this, and this is now quite some time ago, it's around 5 years ago, we thought that we might be able to use approaches that have been developed for bulk RNA-seq around regressing out principal components that would correspond to batch effects, things like surrogate variable analysis, where you find different features that are orthogonal to the biological feature of interest, and you could simply regress them out, and that would be a way of combining data. And so we thought we could do this, but actually there's a problem, and we can illustrate that problem using a really simple simulation. So suppose we've got 2 batches, 2 experiments. In each of these batches, we've got 3 cell types, and these are just plotted on the left-hand side here. And the only difference between these 2 batches is the composition of the cell type. So for example, in batch 1, you might have 10% of cells from cell type 1, and in batch 2, you might have 20%. That's the only difference, apart from this orthogonal batch effect that we've also simulated. Now, when we apply these bulk RNA-seq like batch correction approaches, which I say effectively are trying to regress out principal components that are global or surrogate variables in essence, what you would hope is that you'd be able to map these different populations on top of one another, so blue on blue, green on green, red on red. But as you can see down at the bottom, actually this wasn't possible. And the reason for this is just simply compositional effects. If we make the composition of cell types the same between the 2 batches, but still have a batch effect, then these bulk RNA-seq-based approaches, surrogate variable analysis, etc., they work very well. So it's this compositional effect that we have to control for. And the trouble is, we don't know what the composition of cell types are a priori. And so then my postdoc, Lali Hagverdi, who's now a group leader in Berlin, came up with a quite simple, but I think quite effective approach for dealing with this problem. And that's this mutual nearest neighbors idea that Jessica alluded to earlier. So in this cartoon here, we've got 2 batches, 2 experiments. We've got 3 populations of the first batch, red, blue, and purple. And in the second batch, we've got 3 populations again, red, blue, and gold. And the idea is that in the high dimensional space, or you could also do this in principal component space, what we're going to do is for each cell in the first batch, we're going to ask what cell is it closest to in the second batch. And then we're going to do the same, but in the opposite direction. So again, either in expression space or in principal component space, we could take each cell in the second batch and ask to what cell is it closest to in the first batch. And if they are mutually closest to one another, we're going to define these pair of cells as mutual nearest neighbors. And so some examples of mutual nearest neighbors or MNNs are shown here in panel B. And to give you an example of a pair of cells that would not be mutual nearest neighbors, consider this purple cell here in population W. Now that might be closest to one of these red cells in the second batch, but the same is not true in the opposite direction. So these are not a pair of mutual nearest neighbors. And then having defined these mutual neighbors or MNNs, we can compute a correction vector. So we just use a locally smooth kernel to compute this correction vector so that we don't get such bad discontinuities in the space. And then what you can effectively do is subtract this correction vector and collapse the 2 manifolds on top of one another, with hopefully the goal being that these distinct population, this gold population that doesn't share any neighbors between the batches, will be kept distinct. The problem, if you don't do this, is that somehow this gold population can be collapsed towards some of the other populations. That's, of course, not what you want. And then having done this, you can add more batches and so on. And this type of approach, this MNN strategy, has been adapted or changed or adopted in a number of the more recent methods that have been published for correcting for batch effects in single-cell RNA-seq data. So it seems to have been quite powerful. So simulations show that it works. That's nice. Simulations that you design almost always show that your method works, so it's not very powerful. More interestingly is we could take real data sets here. So this is now a study, 4 studies of the pancreas using 4 different labs, different experimental technologies. And the different studies are indexed by the symbol, and the colors of cell types as annotated in each study is shown by the different colors. And the uncorrected data is on the top left. We could see very substantial batch effects here with, say, 3 clearly distinct green populations. And if you look down at the bottom, this is the attempt to correct these batch effects using these bulk-based strategies, the surrogate variable analysis-like approaches, and they're not doing a very good job. Whereas when you use the neutral nearest neighbors, we seem to be doing much better. We seem to be really collapsing cells that are from the same cell type on top of one another. And that gives us more power to not only identify cell types that are rare, but also better power to detect differentially expressed genes. So this does seem to work quite nicely. And then in practice, we've applied this in a number of different settings. An example that really motivated us was a study of mouse development. So this is now a little bit of a biological example. So we've got 9 sequential stages of early development in the mouse from embryonic day E 6.5 through to embryonic day 8.5. And because of the way these data were generated, we couldn't randomize samples from the different time points together. They had to be collected independently. And so we had very substantial batch effects. So in order to actually integrate these data, we had to apply the MNN approach that I just described before. But having done this, we were able to then obtain a very nice data set that we were able to cluster using, again, graph-based approaches, and then visualize in 2 dimensions. So this is now a much larger data set with around 150,000 cells or so. And you can start from early on in development through endodermal tissues, blood up at the top, etc. And we can visualize this in a number of different ways. I kind of like this ribbon map representation where we've got the time points on the x-axis, on the y-axis are the fraction of cells from each cell type. And it really illustrates very beautifully this increase of diversity of cell types as you go from earlier in development to later, with the majority of cells at the earliest time point coming from the epiblast and all of these colors beginning to appear later on as the explosion of diverse cell types in the embryo becomes apparent. But I think this integration that we did was really just the first thing that you can do. I think that statistically, there are also very interesting questions about what you do when you've integrated these different data sets. And in particular, something that we and others are becoming quite interested in is how do we compare different data sets? So see, I've got normal and disease conditions. I've matched them together. How am I going to analyze these different data sets? And so I'd like to now talk about work that's on the archive, but unpublished at present. Because up until fairly recently, the way that we would do this is that we would typically cluster all of the cells together. And then within each cluster, we would ask, do we have differential abundance of cells from condition 1 or condition 2? And then you could do something simple like a Fisher exact test or some other type of statistical modeling, some fairly simple statistical modeling to ask within each cluster, do I have more cells from condition 1 or condition 2? And this would be, if that existed, then we would see that there's differential abundance of cells from these 2 populations in each cluster. However, especially in development, the definition of discrete groups of clusters is not trivial because lots of processes lie on a continuum. And so this ability to define these discrete groups of clusters is actually quite a major limitation because it's quite hard to do. Where do you draw the line? If I go back here, you can see that, say, early blood development, this is fairly continuous. There are computationally defined clusters, but the boundaries here are rather arbitrary. How many clusters to define, what resolution of clustering should you employ when you're really looking at a continuum? And so what we wanted to do was to come up with an approach that would look for differential abundance of cells between conditions that didn't rely upon cluster labels. So we wanted a clustering-free approach. We wanted that approach to be applicable to data sets with very different topologies, both discrete groups of cells, but also those with complex trajectories, complex bifurcations, for example. We wanted this to be flexible to complex experimental designs. We want to be able to account for continuous covariates. We want to be able to account for confounding effects, for example, kinship relationships between individuals if we get that type of data. And we want to be able to account for the fact that we have biological replicates. So we don't want to merge across biological replicates. We want to use these replicates in our analysis. And so the approach that we came up with for doing this is called Milo. And the goal here is to test for differential abundance, but to do this on k-nearest neighbor graphs. And I'm just going to illustrate this with a little cartoon here. So we've got two conditions, condition A and condition B. And I'm going to assume that we've got single cell RNA-seq from multiple replicates from these two conditions, and that we've mapped these cells onto the same joint manifold. So for the purposes of this illustration, we've removed any batch effects. And then we can construct a KNN graph, and this is just a visualization of such a graph here. And the different colors correspond to whether a cell's in condition A or B. And then what we're going to do is we're going to assign cells to neighborhoods on this graph. So we're going to define a series of index cells throughout this graph that define important regions. So we're going to try and weigh the number of index cells by the density of points. And then within each neighborhood, so around each index cell, we're going to define a neighborhood. And within that neighborhood, we're going to ask whether we have more cells from condition A or condition B. And then, so for each neighborhood, we can construct a simple vector where we count how many cells from condition A there are in each replicate and how many cells from condition B there are in each replicate. And then we can test for differential abundance in each neighborhood using a negative binomial generalized linear model. So similar to the types of approaches that are used for studying differential expression. And the advantage, of course, of a GLM is that we can account for various covariates in the log link function, and then we can control for these when we do the downstream analysis. Now, there is a little bit of a wrinkle here in that these neighborhoods overlap one another inevitably. So there's a bit of overlap throughout the graph. And so when we control for multiple testing, we're going to use the spatial false discovery rate. So not just the conventional false discovery rate, but the spatial false discovery rate, which effectively controls for the degree of overlap between different neighborhoods so that we're able to account for this degree of overlap when we do the testing. Yeah, please, question, Jessica.
So John, I have a question regarding the definition of neighborhood. So how do you define the neighborhoods, basically?
Yes. So in the KNN graph, we can choose, we've got obviously indexes from cells to cells, and we can define how many nearest neighbors around the index cell we want to use for to define the neighborhood. So depending upon the data set, we would choose maybe 30 nearest neighbors, but it depends on the number of replicates you have. If you have a lot of replicates, then you might want to increase the neighborhood size that you use. So there's a trade-off between the number of cells, the number of biological replicates, but effectively we're choosing K and a KNN in the local neighborhood on the graph to define the number of cells within each neighborhood around each index cell. So we're basically taking the nearest neighbors of each index cell, and that's how we define the neighborhood here.
I see. So basically you apply the same K to all cells. It's not adaptive. K is not adaptive per cell.
Correct, yes.
Okay. And then does it matter? I'm just curious regarding the topology. Does different topology of neighborhoods affect the goodness of fit of the negative binomial?
We haven't seen that. So where it goes, where things can go wrong, or where you can, or where you lose power, is if there's very discrete clusters. If you have if you simulate data where you've got 3 distinct groups and there's not a continuum, then basically you're performing too many tests, because you don't, if you've got really discrete clusters, then this type of approach is suboptimal. It works, I mean, as you might expect, it works quite nicely on linear trajectories. We haven't really tested what happens if you massively change the density of points along the graph. I would expect that will dictate the number of index cells, because in regions of the graph that are less dense, where you have fewer points, you're going to have fewer index cells. So you're doing fewer tests. And basically we aim within each neighborhood to have approximately the same number of cells, as I said, that would be fairly fixed in the neighborhood. So I don't think it should have a massive effect. There are problems depending upon different types of topology, but I don't think the fit of a negative binomial is too badly influenced. I mean, I imagine if you had a very sparse region that was very diffuse, then you'd have quite a lot of heterogeneity, and you might have to tune your K so that you're not taking very different populations. And we haven't though simulated a data type like that yet. So it's but that would be something worth thinking about, certainly.
I see, thank you. And I saw that Timothy has a question. He raised his hand. Could you unmute yourself and speak up?
Hi. So I just wanted to clarify, when you were creating your k nearest neighbor graph to do at the beginning of your test, you created it on the manifold that you get after dimensionality reduction, correct?
Correct.
Okay.
Yes.
And I think, yeah, and the other question is like, would hubness play a role in or affect the power of your test? Because like
yes.
you say, okay.
It definitely does. It depends on the, I mean, the goal is, I think a lot of the trick comes in choosing good index cells so that you don't oversample regions where you don't need to. You want to, so we use a trick that was actually something Dana Pear's lab came up with when they were looking waymark points, when they were doing their graph construction approach. And we use something quite similar to try and index useful cells across the graph. I think defining those index cells is a really important part of what we do, because otherwise the multiple testing really does kill you because you just have too many neighborhoods and there's too much overlap. So defining those index cells sensibly is important.
I see.
Okay, excellent. Thank you for the questions. And please, anyone else, do interrupt. So in order to test this, we could do some very simple simulations. So this is just a nice linear trajectory. And again, we got condition A and B. And you can see in this little region of this trajectory where my cursor is now, we've got more cells from condition B than condition A. And that's shown in the density representation down at the bottom. And then the way that we're going to identify whether a particular neighborhood has differential abundance, we're basically just going to do this GLM test in each neighborhood, do the multiple testing correction, and then the neighborhoods that are significant that you're going to see in the next couple of slides are indexed by the different colors. So darker colors indicate that we have significant differential abundance, and indeed we pick out the neighborhood that we simulated as differentially abundant here. So how do we benchmark the different approaches? Well, we can either construct a KNN graph using synthetic or real data. This is a real data set here. Then we're going to generate a condition probability. So we're going to assume we have two conditions. And then in most of the graph, we're going to have an equal chance of sampling a cell from condition 1 or condition 2. But we can then identify a particular region here where we've got a higher probability of sampling cells from condition 1. And we're going to assume that cells in regions of differential abundance are going to be truly differentially abundant if the probability of that cell coming from condition 1 is more than 0.6. Jessica.
I just want to say that there's a question in the chat box from Liang.
Oh,
he said, or he or she asked, so you choose index cells biologically?
No, they are chosen mathematically.
Thank you.
Excellent. And so having generated these simulated data, then of course we can then within each region, we can assign cells to the different conditions on the basis of these probabilities, and then across different replicates within each of the conditions as well. And then we did this across a variety of different settings with different simulated fold changes within each region. And we were able to show that in general, the approach that we developed, Milo, was fairly good at controlling for false positives, so it controlled the FDR pretty well. And as you might expect, as the fold change, as basically the differential, the strength of the differential abundance between the conditions increases, you have more power to detect true differences. By contrast, if you use a graph-based approach, so that's basically here in the middle, that's Louvain clustering, and then within each cluster, we're going to test whether we have differential abundance using a GLM. You almost always will detect everything that's different, but you have no control at all over false positives. So basically you're saying there's differential abundance everywhere because this fine-grained clustering that's needed to get down to this resolution is just defining far too many neighborhoods. And other approaches that we compared it to seem to have also some particular problems either with power in the case of this approach here, or basically just everywhere in this approach that was designed for proteomic studies. And then we can apply it to real data sets in practice. So this is an example of a data set looking at cirrhotic and normal liver cells. So we've got two conditions here. So this is from a group in Edinburgh, and they did single cell RNA-seq. It's plotted here on the left-hand side. The cell types are indicated by the different colors. And then you can see the labels of the cells, whether they're from cirrhotic or uninjured livers here. And you can already visually see when you look at panel B, that, for example, in this region here around mast cells, we have many more mast cells in the uninjured than in the cirrhotic condition. And indeed, we're able to identify this region and pull it out directly from the data, as well as other more subtle regions where there's differentially abundant populations of cells between the normal and the diseased condition. So I think that this type of approach is kind of nice. And I think that actually there's going to be quite a lot more use for these approaches where we are trying to compare normal and diseased cells between different conditions, because I think this is going to be, with the generation of things like human cell atlas, a very obvious challenge is to take, say, the normal lung data from it, and then to map on samples, say, from individuals with COVID, and to work out what's the difference in the cell populations between these normal and disease conditions. So at the moment, there are not so many methods for this kind of differential abundance testing out there, but I think that this is probably going to be an area where there's going to be quite a lot of work in the next few years, and also incorporating genetic variability, for example, into this analysis as well. So kind of the type of work that we do fairly frequently with EQTLs, you can also think about incorporating that genetic relatedness into these differential abundance models. And as I say, that work is on the bioRxiv. So if you're interested, please go and have a look at that paper there. It was led by a fantastic student in my lab, Emma Dan. Okay, so that's one type of data integration. That's an example of horizontal integration, where we're using the genes as the different features for the anchoring. But there are also vertical integration, and this is something that's more akin to these multi-omics methods. So some of you might be familiar with the 10x attack RNA platform, where from the same cell, you're able to profile gene expression and chromatin accessibility. But there are also other techniques. For example, there's SCNMT, which my colleague Wolf Reichert of Abraham came up with, where you can measure gene expression, DNA methylation, and chromatin accessibility in the same cell. And of course, there's CytSeq, where you can measure antibodies and RNA expression, and a variety of other approaches. And here, when we're thinking about integration, it's not the features that are the anchors, it's the cells that are the anchors, because the cells are the thing that are in common across these different multiple molecular layers. And so we can think of integrating these different molecular layers here, methylation, expression, and chromatin accessibility in the little cartoon, by using the cells as anchors. And the approach that my student Ricard Archelaguet came up with, who I co-supervised with Ollie Stiegel, he just recently graduated, was a flavor of factor analysis. And he termed this approach multi-omics factor analysis. Effectively, it's a multi-view factor analysis model. And here, each view is a different molecular layer. So in this context, one view could be RNA expression, a second view could be DNA methylation, and a third view would be chromatin accessibility. And then the cells, as I just mentioned, are going to be the anchors. And what you want to do in a multi-view factor analysis, so I'm sure you're all aware, is that instead of decomposing the factors that explain variation in just one of these views, we want to define factors that explain variation that can be shared across views, as well as variation that can be common to just one view. There are a number of subtleties about this model and challenges in implementing it. One of the major ones is that the number of features differs quite a lot between views. The number of genes, for example, is quite fixed. There are around 10,000 genes that might be expressed across all the cells in a single cell RNA-seq experiment. But the number of loci in the genome where you can measure DNA methylation, of course, is in the millions. So you've got really very different scales of data across these different views. And so what we would suggest is that you want to define the features that you're going to study, the DNA methylation features, quite carefully. So you might want, for example, look at methylation in promoter regions, because if you have really big imbalances between the number of features in the different views, that can mean that the view that has more features will dominate the decomposition, and that's not a good thing at all. So that's one challenge. And another challenge is that, of course, the type of data, the statistical distribution describing each of these data sets can be quite different. RNA expression is counts, but DNA methylation, in principle, at least, is binary data, it's 0-1 data. So we've implemented a version of this factor analysis where you could have different distributions. So you can have, say, a log normal distribution for the RNA expression. You could have a binomial distribution for the DNA methylation. The trouble is this comes at the expense of more computational time. It's much more efficient if we can make a normality assumption everywhere. So it really depends a little bit upon the size of your data set. If you have a smaller data set, it's possible using these distributions and some stochastic gradient descent type approaches to fit the factors relatively well. But still, in many contexts, actually, you'll find that just making a Gaussian assumption works actually reasonably well. And that's what we have done in practice. And if you're interested, those papers are described in a little bit more detail down at the bottom here. So these 3 papers that Rickard worked on. But what I thought I would finish with was something a little bit different. And that's the integration of another type of data. And that's an integration of single-cell genomics data with spatially resolved data. So all of the data sets that I've described up until now basically start with your cell type of interest. It might be the embryo, and you dissociate all the cells and you lose the spatial context. And that spatial context matters because physical constraints, where the cells are in the embryo, the signaling environment, is really going to dictate what they do. So you can infer some of these features from the high dimensional genomics data, but actually there's only so much you can infer without that quite precise spatial information. Now, there do exist in situ databases, especially in the context of development, where maybe people have probed the expression of 2 or 3 genes within the same context, but typically there's an inability to measure expression at single cell resolution, and you really can only assay 2 or 3 genes at any one time. But similar to the single cell genomics revolution, there's now increasingly becoming a spatially resolved single cell revolution, I think, where we're able to profile expression, but not just expression, other molecular markers in cells within their spatial context. So approaches such as highly multiplexed RNA-FISH from the lab of Long Kai at Caltech and also Xiaowei Zhang in Harvard are one such strategy. And here, this is effectively a multiplexed RNA-FISH experiment where instead of profiling the number of molecules of a single gene in a cell, you're able to use a clever combinatorial indexing strategy to profile the expression of dozens or hundreds of genes within a spatial cell. At a slightly more coarse resolution scale, there's the Visium platform for 10x. Here, basically, you take a tissue section, you hybridize it onto an array that's spotted, and each of these spots has a different barcode, and that indicates the position on the array. Basically, you then you hybridize the sample to the array, and then you elute all of the RNA molecules off the array, and they are indexed by a particular barcode, and that tells you where that RNA molecule comes in a spatial context, as well as other more complex approaches that are increasingly, though, being adapted, including in situ sequencing for the lab of Mats Nielsen at the SciLife lab in Sweden. And again, just as for bulk RNA sequencing and single cell RNA sequencing, these new types of data motivate the development of new computational methods. So for computational biologists, it's almost like there's endlessly fun things to think about. One of the challenges, which is non-trivial, is that these are not unbiased approaches, at least the seqFISH-like strategies, which is the one I'm going to talk about. Here you have to decide what set of genes you're going to study. So you have to decide, what am I going to input and what am I going to probe? And so I'm going to give you one example in the context of early development. And the way that we designed the set of genes that we wanted to probe here was that we took this atlas that I described earlier, this early developmental atlas in the mouse, and effectively we built a fairly simple classifier to identify the set of genes that could best delineate each of these different cell types. But in addition to identifying a small number of genes that could mark each cell type, we also wanted these genes to be lowly expressed, because if they're too highly expressed, it's difficult to resolve the different dots when you do the microscopy. And in the experiment I'm going to talk about, we probed in total, after doing this analysis, the expression of 351 genes. So what exactly did we do? Excuse me. Well, at a particular stage of development, this is embryonic to 8.5 to 18.75. For those who are interested, this is around the 8 to 12 somite stage. we took 3 independent mouse embryos, and the experimentalist Tim Lohoff went from Cambridge to California to take sagittal sections through these embryos. And these are the 3 sagittal sections here on the right-hand side. And within each of these red boxes, which are called a field of view, we're going to probe the expression of these 351 genes using this fluorescent in situ hybridization strategy. So what does the data look like? Well, if you zoom in now on one of these fields of view here, and this red square indicates one field of view, then this is kind of what it looks like under the microscope. And in this top left panel, we're staining for the expression of a number of cell membrane markers, so different cadherins that mark the boundaries of each cell. And that motivates the first statistical downstream analysis problem, which is to basically use these membrane stains to segment the image into individual cells. And so here we used a machine learning classifier called Elastic, developed by one of my EMBL colleagues, Anna Kreshuk, where we trained using the membrane stain and used that to then basically segment the whole image. And the segmented objects are shown here on the top right. And then within each of these segmented blocks, we can zoom in still further on it down at the bottom. And within each of those blocks, we can count the number of dots that correspond to each gene. And so within each of these segmented regions, we're able to count and measure the expression of 351 genes. Now, this hides an awful lot of work. And I think that in particular, segmentation, accurate segmentation is really difficult. We are able to take advantage of a membrane stain here that really helps. That's not always going to be possible in many settings. And so improved approaches, statistical approaches for segmenting images and to identify real cells is really going to be important. But having generated that segmentation mask, we could then look at the expression of individual genes in every cell. And what we have now, of course, is a gene expression matrix where we've got genes by cells, but each cell is now indexed by a physical location. And we can plot the expression of individual genes. So here's the expression of 12 genes, for example, genes that mark the heart in these first 2 panels, endoderm genes. So this is the gut tube down at the bottom right. But the real power of this approach comes not from plotting the expression of single genes, but by combining all of these genes together. And the first thing that we wanted to do was to take each of these segments here and to assign it a cell type identity on the basis of the set of genes that are expressed in it. And the way that we did that was to adapt the neutral nearest neighbor strategy that I described earlier. So we're going to use the 10x reference as one data set, and we're going to use the spatially resolved data as a second data set. And basically, we're going to map them onto the same manifold here, not taking any account of the spatial information. But having mapped the spatially resolved data onto the 10x atlas, we can then use that atlas and ask what are the neighbors of each spatially resolved cell in the 10x atlas, and use that to assign each cell in the seqFISH data a cell type identity. And what does that allow you to do? Well, it allows you to overlay on these embryos and the color of the corresponding cell type. So what we've done is we've taken single cell RNA sequencing data, integrated it with the sequential FISH technology data, and then used that to assign each cell in a spatial context a cell type identity. And why did we do this? Why didn't we use a random field-like approach just to cluster the spatially resolved data? Well, in essence, it's because we've only got 351 genes here, whereas we've got tens of thousands or 10,000 genes in the single cell RNA sequencing data. And we reasoned that that would give us a stronger ability to identify rare cell types and to better define them. So by integrating the data at this point, we've reasoned that we could get a better identification than just a straight clustering of this data type on its own. And actually, we were able to show that this approach, this joint approach, does indeed give you more power when it comes to assigning cell type labels. So there's really value in integrating technology at this time point. So we don't have to be restricted, though, to these 351 genes. Once we've integrated the data sets once and mapped them onto the same manifold, we can impute mRNA expression for all genes. So basically, for each seqFISH cell, we map it onto the reference as described. And then using the set of nearest neighbors, again, we can take the average expression of each gene amongst that set of nearest neighbors in the RNA sequencing data set and use that as the imputed expression of that gene in a spatial context. We can test this via simulations, but more powerfully, I think, we can compare imputed data, which is down at the bottom for 6 genes here, with independent validation data at the top here. And this is really satisfying because you can see that both for specific marker genes like NKX25 that marks the heart, we're getting very similar patterns of expression from our imputed and real data, but also for positive control genes and negative control genes, we're getting really quite nice overlap in expression patterns, suggesting that this imputation strategy is both specific and sensitive. So finally, just in the last couple of minutes, how can we use this? Well, we can take this imputed data, we can zoom in on a particular region of the embryo. Here, this is a particular region of the brain that we've further defined into subcell types. And then we can computationally dissect this region into 2. So we can define the midbrain hindbrain boundary here, which is indexed by the expression of GBX2 on the left and OTX2 on the right. And then using the imputed data, we can ask what genes are differentially expressed between the 2 conditions. And this just gives you the power to identify new marker genes, and they can be motivating examples for downstream biological discovery. Okay, so I've spoken for about 45 minutes, so I'll finish there. I think that we're at a really exciting time in computational biology because we're getting all of these complex multimodal data sets indexed either by cells, by features. Sometimes in some cases, there may not be any features at all overlapping between different data sets, neither cells nor any of the molecular features. And that's, I think, a very interesting thing to think about how we integrate these mosaic kind of data sets where there's maybe no overlap. Do we impute, or do we, for example, identify factors that might be shared between the different data sets, correlate them and use them as anchors? I think that's very exciting. I think that this integration of spatial data together with this more classical molecular genomics data is going to be really important. And the next step, I think, in this is going to be the combination of temporal data, how these cells move through time. So I think over the next few years, the ability to integrate spatial data with temporal resolution, for example, using nuclear tracking assays as developed very beautifully by Philip Keller and Kate McDowell at Janelia, is going to be really exciting. And once we have these data sets, we can then begin to think about how we can use them to really understand fundamental processes, for example, in the context of early development. So with that, I'll close. I'll thank my lab. I have a wonderful lab. I'm not going to single any of them out, but they've all contributed tremendously to the work that I presented, as well as ex-lab members and members of other labs I've worked with over the years, as well as wonderful colleagues, especially in the gastrulation work that I talked about, Wolfreich, Bertie Gutkins, Long Kai in particular. And although she's not mentioned here, I should also acknowledge my good friend and colleague Sarah Teichmann at the Sanger, with whom I co-supervised Emma, who worked on the differential abundance work at the very beginning. And with that, I'll close. I've answered a few questions already, but I'm happy to take any more questions that you might have, and also to answer questions over email if people have other things that they're interested in. And thank you very much for your attention.
Thank you very much for your wonderful talk, John. And we have a question from the chat box from Amelia. And greatly interesting talk. Thank you. My question re metabolomics data. What will be the contribution utility of metabolomics data in such context? Thank you.
There will be a lot of utility of metabolomics data, and especially spatially resolved metabolomics data, which I think is becoming increasingly not trivial, but becoming possible, I think. And I think that one of the limitations of a lot of the work that we've done to date is that RNA is the thing that's been measured. And obviously RNA is good, but it's only one molecular layer. And the ability to have more molecular layers is going to give you just more insight. I think a really interesting and not so trivial challenge is the lag between these different molecular layers. So if you think you've got multiple things measured in the same cell, so it could be RNA expression or, RNA expression, the RNA expression, DNA methylation, and you've got them ordered along developmental time, do they change at the same rate? What comes first? And so understanding what that lag is, because there's going to be some degree of correlation you would expect, but what is the lag in the correlation? Does that change across developmental time? Does that change across different developmental systems? Probably it does. And so I think these models that are trying to look at various autocorrelation strategies and other strategies for locally weighted correlations between different molecular layers in developmental time are going to be really interesting as well to think about. So I think that there's definitely more work to do in that context. But basically, I think that more data gives you more information. I mean, Amelia, do you have a particular context where you think that such data would be useful? Okay. So.
All right. Thank you for your answer, John.
No worries.
There's the Money from Money. She has a question. Is there a list of references in one place?
I will send that to Jessica and she can forward it onto the list.
Oh, great. Thank you. I think we'll try to post that on the WNAR website along with your slides. Yeah.
Of course.
And then Yiliang Zhu, you said that you choose index cells statistically. Is it because of the lack of biological input or evidence?
So I guess so you could choose them. You could choose them biologically. The trouble is then it's not very robust, right? Because it depends upon one person's interesting index cell might not be the same as the interesting index cell of the second person. So ideally, to make the work reproducible, you want to basically have an approach that's agnostic to the individual investigator. And so we want to choose those cells that are, we think, representative of the manifold. So basically you want to, there's a different set of ways of doing this. You want to choose a set of cells that represent the high dimensional manifold. So basically you're going to want dotted cells along each part of the manifold. You're maybe going to want more index cells where you've got a bifurcation. And you want to choose those cells that basically the minimal set of cells that will allow you to reconstruct the manifold in essence is what, at least in my mind, that's probably what you want to be having here. And that will give you the ability to cover the whole space without having such a multiple testing burden. It also means that you're not biased to an individual investigator. Does that make sense, Julian?
Yeah, I think it makes sense to me.
Excellent. I hope so.
Then Amelia has another question. And
yeah, I think, sorry, I was on mute. I think it makes sense. But I think, you know, we choose index events quite often in health and clinical research. So I think you did allude to the challenge of using biological evidence to support choice of index cell. But I guess when there are clear definition, but that makes sense, I think, you know, biological criteria makes sense. That leads to my second question, that is, you have alluded to that already. I thank you very much for that, for doing that. That is, statistical criteria could also be multidimensional. And there's not a single criteria say, this is how I do that, but you talked about, you know, some of the criteria. So that was my second question. You know, mathematically, without any criteria, it's a wide spectrum. Each cell can be an index cell, and I can choose arbitrary just one cell as an index cell. Then in between, I have many choices. So if we could elaborate further, that'd be great. But I think you already touched upon that. Thank you.
Yeah, so I think that one context where you would clearly want to choose, or where you might want to choose index cells would be discrete clusters. I sort of mentioned this in the presentation, but when you have very discrete groups, then in some sense, an index cell could be the cluster centroid, right? And then basically you would consider everything in the neighborhood of that cluster centroid would be the different populations. And that's going to give you the most power to detect differential abundance if you are certain that that is one cell type cluster, right? So in that context, you would probably not want to use, frankly, the type of approach we had, simply defining the clusters and then using a Fisher exact test or multinomial regression. Just simple multinomial regression is probably going to do you just as well. And you're going to have much more power because you're doing fewer tests. So I think that if you have discrete clusters, that would be one case where you could use the type of approach we have to define index cells, but I think actually you would lose power in the analysis. So as always, you should say there's this spectrum of human intervention that we know that these are the clusters, this is what we care about, through to agnostic analysis. And I think the more complicated and the more continuous the trajectory, you want to be more and more agnostic and let the computational method in some sense do the work, because otherwise you have this lack of reproducibility from individuals.
Thank you. And I think Amelia has left, but she left a question here. And variability across different gene sets as well. Very interesting. Thank you so much. Right, I think it's just a follow-up from her.
Yeah, I think so.
All right, so then Laura, thank you for the great presentation. Can you please clarify what is the benefit of the segmentation and extracting information of each cell versus just spatial transcription analysis by regions of interest? Thank you.
That's a great question. So you could, in principle, just index each dot, each RNA molecule by an XY coordinate, and use those as the set of features. And then you could look for patterns that don't require you to do any segmentation at all. I think that one of the motivations for the segment, so it's absolutely a valid approach. I think that the hope would be that there's additional information that you get from the segmentation. You could potentially think around things like cell morphology, cell size, other features that you could use as covariates to try and understand what's going on. And also, sometimes if you don't define cells, it's quite hard to define boundaries between cell types. And at least early in the embryo, there are quite clear boundaries. And they, of course, are going to be defined by cells. You're not going to, it somehow would be a bit weird to draw a boundary that cut multiple cells in half, because half a cell generally isn't going to be cell type 1 and the other half cell type 2. So if you have that boundary information and you want to then segment the cell or segment the image into different regions, it somehow feels intuitive to do that segmentation first so that you can then really define that boundary properly. But certainly for exploratory analyses, or more unbiased analyses, there actually is no need to do the segmentation. You're right. You could just use, you could basically just voxelize the image, and within each voxel, ask how many counts there are from each gene and look for correlation patterns that way. That would be another way to do it. And actually, that's kind of the type of analysis that the Visium platform, the 10x platform, allows you to get at, because that's the array-based approach where you hybridize the tissue section to the array. And there, each spot corresponds to data from multiple cells, not many, but 5 or 10 cells. And there, what you're looking at basically is patterns where you don't need to do segmentation. And you absolutely could do that in the context of the RNA-FISH data. It's just that for some analyses, say defining cell type boundaries, it really does make sense, I think, to want to segment the images first, because those boundaries are defined by the cells themselves rather than kind of cutting a cell halfway through. I hope that makes sense.
Yes, thank you.
Thanks, Laura.
Thank you, John. And I have a question regarding the last part of the spatial data when you do differential analysis. So when you try to compare, let's say, two tissue slices from two conditions, so would you need to like rotate them in some way so they're aligned, or we should call them registration?
That is a great and horrible question. So in essence, we don't at the moment. We do not really integrate the images. And there's a number of reasons for this. One, they're not, although it's the same stage of development, and the aim is to cut a similar section, there's just differences in the angle of the cut.
Right.
There's differences in how it's processed. The stage of development will also differ slightly. That makes it really hard to map these different images directly on top of one another. And I think this is true for a lot of spatial transcriptomics type data. And it's certainly going to be true in the context of cancer. People are applying spatial transcriptomics in cancer. And of course, data from 2 breast cancer samples is not going to have the same spatial organization. It's going to have common features. And I think it's these factors, these features that you're going to use to map the data sets together. So for example, you could identify things like how near is a cell to a blood vessel? How near is a cell to the tumor margin? It's going to be those sorts of things that we're going to use to combine the images. I don't think we're going to create a common reference, or maybe we will, but it'll be more like a generative reference where you use machine learning to identify a set of features, and then you simulate a reference using a generative model, for example, and that will be somehow the reference data or the reference spatial data that you might use moving forward. But I don't think it makes, at least my intuition, and I'm happy to be proved wrong though, is that it doesn't make sense to try and map these different types of spatial data on top of one another. Initially, that was actually what we thought we were going to do, but we just looked at the data and there was just enough heterogeneity between the different samples that it just didn't feel like it was going to be possible to do this very well. So we gave up on that. And I do think now that maybe these abstracted features that we learn from the data are going to be how we combine the different spatial omics technologies, for example.
Thank you very much. I think that makes a lot of sense.
Pleasure.
Okay, so do we have any other question for Dr. Marioni before we finish today's webinar? All right, so if there are no more questions, let's thank John again for his wonderful talk. And,
thank you very much.
And also, we are really looking forward to your future work in this field.
Thank you so much, Jessica. Thank you, everybody, and have a lovely weekend all.
You too. Bye.
Bye.
Thank you.
Bye. Bye.